The second sort of global screening approach was from Gina DiNicola, a grad student who did a proteomics screen, looking at RAS mutant versus RAS wild-type cells, doing a very standard mixing assay, and she came up with a group of proteins involved in redox regulation. When this happened, I was not particularly surprised because I carry around the same notion that was actually mentioned in this conference several times. That is that oncogenes activate reactive oxygen species and cause an oxidative stress inside cells. And so when Gina actually did the direct experiment of turning on RAS in cells and measuring reactive oxygen species, I was fairly startled by that. And so that's shown here. When you turn on RAS, you get less ROS in cells. And these are in mouse fibroblasts. They could also be found in primary ductal cells. And that's the opposite result of what you would find by reviewing and reading the literature. When you overexpress RAS, the opposite happens. You get more ROS, be it HRAS or KRAS in the oncogenic form. And if you look at oxidized DNA as your end product, instead of using these dyes which approximate hydrogen peroxide, you get the same result. So overexpressing RAS, you get more oxidized DNA. Endogenous KRAS reduces the amount of oxidized DNA in that cell. It's mirrored by the reduction of glutathione, where in an endogenous system you have more reduced glutathione compared to overexpressing, where you have more oxidized glutathione. So where does ROS come from? How is ROS managed? This is, again, a topic that many of you will be familiar with, but just a brief reminder, the mitochondria really is the source of most ROS produced by cells, but there are other enzymatic cascades and non-enzymatic cascades which can result in ROS accumulation. And so we examined these in our cells, and we didn't note any particular difference in ROS production when we turned on oncogenic RAS endogenously. And so we looked at the metabolism arm, which is responsible for neutralizing and for converting ROS. One particular pathway which we honed in on was the glutathione thioredoxin system, which is controlled to a large extent by a transcription factor, NRF2. And in fact, NRF2 appears to be targeted for expression by turning on the endogenous oncogenic KRAS allele, shown here by chromatin IP, where you have increased target gene expression, as well as just by TACMAN PCR looking at a variety of products. This appears to be important, because if you knock out NRF2 and then turn on RAS, the cells are not happy. They do not grow very well. And you can do this either constitutively or acutely with siRNA. By using pharmacological inhibitors and genetic approaches, Gina was able to hone this down into the MAP kinase cascade by, again, reverting the effects using a MEK inhibitor similar to one that you heard about from Ed. This one's produced by AstraZeneca. In vivo, this pathway appeared to be active. Shown here in the human, a downstream product of NRF2 and QL1 in a pannin compared to a normal ductal cell in the human pancreas. And again, you can actually stain for levels of oxidized DNA using antibodies against 8-oxo-DG, and the levels are low in the RAS mutant pannin and higher in the normal ductal cells nearby. This is an interesting transcription factor in that it and its binding partner, KEAP1, have been found to be mutated in a significant fraction of human cancer recently, with the suggestion that this is a pro-tumorigenic mutation. Mutations have been found in head and neck cancer, lung cancer, occasionally in breast cancer. The hotspots are shown here for the mutations. This concept that RAS may be a new way to activate this pathway by increasing the transcription of NRF2 was new, and so we wanted to know in pancreas cancer what was the mutation frequency of either NRF2 or its binding partner, KEAP1. And so we sequenced over 100 samples with Scott Kern at Johns Hopkins of human pancreas cancer. The vast majority contained KRAS mutations, though not all. We found no NRF2 mutations and 3 KEAP1 mutations, but only 1 in a sample that had a mutant KRAS, showing this overlap was very infrequent and statistically obvious. We then, being mouse geneticists, having trained here, did some experiments which are simple to summarize but take a bit longer to do, where we asked in the pancreas and in the lung, would this have any impact on tumorigenesis? And the punchline is that the answer is yes. It did not totally obliterate the ability to initiate or progress, but had a very marked impact. And so here's the NRF2 knockout. You still see some pannins compared to NRF2 wild type when you turn on RAS in the pancreas. But the number of of pannins is low, and the proliferation index of these pannins was only 25%, that of control. The target genes were now missing. These cells were now more oxidized, and a very similar result occurred in the lung, where there was a lot less disease in the lung, looking at any of the subtypes of the pre-neoplasia of lung cancer. And again, the proliferative rate was very much reduced, with a big survival advantage. Again, suggesting that this is another pathway downstream of RAS which may promote cancer by causing a reduction in the intracellular milieu. Well, you can't knock this gene out in humans, but the way we could go about it was to use pharmacological methods, and I already told you that it appeared to be a MAP kinase cascade target. And so again, using the same MEK inhibitor, Gina was able to show that you could lose target gene expression. And in a different way of doing the same experiment, she was able to deplete glutathione from the whole mouse and show by doing so that the proliferation went down in the pannin compartment, but was unaffected in tissues that didn't carry a RAS mutation. And importantly, in pancreas cancer models, you could actually extend survival of those models just by depleting glutathione. So again, this other new pathway downstream of RAS that was found through a proteomic screen, which appears to be relevant in vivo, is shown here, where RAS is not causing an increase in ROS, but actually the opposite, causing a decrease in reactive oxygen species, which could be targeted again by a MEK inhibitor or BSO, but again, leading to potential applications in the prevention space or even in invasive cancers. And Gina was assisted by her collaborators at Penn and at Hopkins on this. And this brings me to the very last point I'm going to make, which is you heard from Ed about some exciting doublets that you could use in patients now to actually try to help folks with lung cancer and other types of cancers. We were the preclinical arm at looking at one of those such collaborations, looking at the AstraZeneca MEK inhibitor and the Merck AKT inhibitor, which themselves as monotherapies were very good at inhibiting signaling downstream of those pathways. And we compared that to knockdown of KRAS in our mouse pancreas lines. And when you knock down KRAS, the cells don't care if you grow them in 2D, but if you grow them in 3D, they don't grow. So this is a similar observation that René Bernard's had in that first paper in Cancer Cell describing knockdown of an oncogene for the first time. Very similarly, when we use the drugs in 2D, they're not that active at inhibiting proliferation of cells compared to when you look in soft agar or in suspension. And so then the obvious experiment is shown here, where the monotherapy arms had very little effect, but the combination did have a big impact on survival, suggesting that there is a path forward. So again, I'd say based on these last few experiments, I think that these two pathways, the PI3 kinase and MAP kinase cascade, are going to be relevant moving forward. And these last two slides are a bit for levity, but a bit for seriousness. And so when I started in England, we started a cancer center, not unlike what you're doing here in the Koch Center. And it was initiated by the royalty and Bruce Ponder, who's the director of our facility. And she came and saw me, just like you came to see me. And I had some posters. And they told me I had 2 minutes to talk to her, but she stayed for 10 or 15 because she had a lot of questions. And I tried to answer all of her questions. And in the end, she said this: "This will take some time." But I didn't like that. And so I said, that's why I hired such a young staff. And you are the young staff, and so I wish you success in setting up your new facility. Thank you.